Okay, moving on to viral replication. Um, okay, there we go. Viruses are obligate intracellular parasites, which you already mentioned. They don't have a lot of genes, so the genes that they possess usually encode the information on how to make capsid or any other subcomponents. Also, they have genes which will trigger the host cell to replicate and biosynthesize just the viral particles. And also, some of the gene will kind of govern the movement the virus inside and outside of the cell. Uh, the example that we are going to talk about is the example of the bacteriophage. That's a virus that is attacking bacteria. Okay, so it's like bacteria pathogen, like pathogens have their own pathogens. This virus is kind of like it has a more complicated structure, like I mentioned, compared to some of the viruses which are attacking eukaryotic cells. So there is a capsid, and inside it's either DNA or RNA. There is this like neck-like structure, a base where you will have hydrolytic enzymes to puncture the cell wall of the bacteria, and then these leg-like structures which are used to attach the viral particle onto the bacterial cell. A lot of text, let's just go straight through the picture. So what is really happening in the stage 1, which is attachment, is that the virus will use these leg-like structures to attach itself to the bacterial cell wall. In stage 2 penetration, what will happen is that the hydrolytic enzymes that are located right here in the base will be used to kind of puncture a hole through the bacterial cell wall. And now when that happens, the DNA will migrate from capsid through the neck region and then inside the bacterial cell. Now, even though this is a very good representation, I don't like because there is a huge thing that is missing. So between stages number 2 and 3, there is a huge chapter that is missing in this picture, which is extremely important, and that is this picture right here. Okay. This circle, that's the viral DNA. So what needs to happen in this stage is that viral DNA has to be incorporated into the bacterial genome. So here you can see this is the viral genome and this is the bacterial genome. So the viral genome will be incorporated into the bacterial genome. So this is an extremely important step that has to happen. Otherwise, the DNA of the virus will not have access to all the enzymes which are necessary to replicate and make copies of the DNA and assemble all the subunits. So once this happens, then all the enzymes will rush to this site and they will start to make RNA copies, and then they will start to biosynthesize the subunits of the bacteriophage. So now in the process of the maturation, all the subunits will come together to form a complete virus. When you have millions of copies being made inside the bacteria, they will use the same hydrolytic enzymes to lyse the bacterial cell from the inside, and then you will have millions of copies coming out of the cell. This graph, this gift is representing this very nicely. So you only need one virus to inject the DNA inside the cell, and then millions of copies will be, bursting out. And of course, in this process, the bacterial or any cell will die. Okay, so when you puncture a hole in the balloon, all the water and cytoplasm and all the organelles and everything will just like kind of leak out, and of course the cell will die. Here you can see some of the genes inside the virus. So these are genes to how to make those capsid in the head, how to make those tail proteins, some of the genes for the recombination. These are the genes that repress the expression of the host genome. Right here. So we have a couple of genes there, and then some of the late control when the virus is about to venture out. This cycle that I just explained, it's called a lytic cycle. And in this case, when the lytic cycle happens, the cell will die. So you have a virus invading a cell, injecting its DNA, millions of copies are being made, and they just venture out and the cell dies. So that's the lytic cycle. There is another cycle called lysogenic cycle. Now this one is a little bit tricky, because what is going to happen is that the viral DNA will be kind of attached to the bacterial genome, so it's going to merge with the bacterial genome, but it's not going to be expressed. So it is going to remain dormant. So this purple circle, that's the viral genome. So it is going to allow bacteria to multiply many times up until this is not a fully functional viral DNA, and it's called prophage. So when it becomes functional, it is going to go into the lytic cycle. So the question for you, so the critical thinking question for you guys will be to think about, why does the virus decide not to kill the bacteria, but to kind of remain dormant and, kill the bacteria a little bit later? Like in the cartoon, I'm not going to kill you now, but I will kill you later. So why is this happening? So try to think about that, and then in the later presentations, I'm going to give you an answer. So these are lytic and the lysogenic cycle. Now this is a story about magical properties of a river in India called Ganges River. And considering how dirty it is one of the cleanest rivers that there is. And yes, that sounds very confusing, but there are, millions of people that live around Ganges River, and there is a lot of trash. So usually what you expect in this type of environment is a lot of eutrophication. So a lot of nutrients that are being input because of human activity. So when this happens, you see a lot of outbreaks of cholera, of food poisoning, a lot of E. coli from sewage. But we don't really see this in Ganges River. So even though there is a lot of human trash, Ganges River is remaining to be very clean, and we do not see those type of outbreaks. It's actually quite the opposite. People come to Ganges River and then they bathe in their water thinking they will be cured. So researchers have been wondering for a very long time, why does this happen? And what they discovered was that there is a bacteriophage, so a virus that is constantly killing off those dangerous bacteria. So, the more human impact there is, the more bacteria there will be, and then that means more viruses will attack and kill off the bacteria. So this bacteriophage is kind of keeping the number of those dangerous bacteria that would otherwise cause food poisoning. They're keeping them in check by, constantly killing them. And this was a very recent discovery. Yeah, like if you have time, I mean, especially now that you're under quarantine, watch this movie Alien: The Eighth Passenger, which is a very similar to a lytic cycle in the virus, where you have this alien that lays eggs inside the humans, and then a new alien is coming out. So if you don't have anything to do, it's a cool movie to watch. It's a little bit older movie, but I mean, it's a Gorny Weaver, so I would suggest you watch that.